Nature has introduced great variety into the landscape, but man has displayed a passion for simplifying it. Thus, he undoes the built-in checks and balances by which nature holds the species within bounds. Rachel Carson.
A who's who of pesticides is therefore of concern to us all. If we are going to live so intimately with these chemicals, eating and drinking them, taking them into the very marrow of our bones, we had better know something about their nature and their power. Also, Rachel Carson.
Welcome to The Wandering Naturalist, a Three Rivers Park District podcast. I'm Angela, she, her, a wildlife biologist with Three Rivers Park District.
And I'm Brandon, he, him, an interpretive naturalist with Three Rivers Park District. Three Rivers Park District manages over 27,000 acres of parks on lands that are the ancestral and contemporary homeland of the Dakota people in suburban Hennepin County and surrounding areas.
So join us as we wander from park to park and discuss your stories of the past, the nature in our present, and how they have shaped our parks.
Come wander with us,
The Wandering Naturalist.
So today we're talking neonicotinoids and their
mouthful.
environment.
We just appreciate that is hard to say.
It is hard. I'm going to be mostly saying neonics for the episode.
Exactly. So that's a challenge for our listeners, hopefully to challenge themselves with neonicotinoids.
Yeah,
breaking it down helps me.
That is a great way to do it. Break it down in those little tiny elements. It felt fitting to start with some quotes by Rachel Carson. To me, Rachel Carson is maybe the most famous person I know of who really sounded the alarm for DDT and its effects on the environment, especially in her book, Silent Spring. And so as we talk about pesticides today, let's go back to Rachel. Let's read a few of her quotes because I feel like they apply. I don't know about you, Angela.
Yeah, it was definitely interesting to see that, she has so many specific quotes about this, like way early on.
It was hard to choose. Yeah. It was really hard to choose. As I was researching this, because neonicotinoids have been talked about, at least in the naturalist world, around bees and whatnot, for a very long time now, it feels like. Years and years and years. And I thought I knew quite a decent amount about them, but there was still a lot I learned. Like, apparently they were first commercially available in the U.S. in 1991. I had no idea they were around that early. They are currently the most used group of pesticides in the U.S. for corn and soybean and other things, but corn and soybean fields really use a lot of it. And I think the thing that surprised me the most was that a lot of the flea and tick prevention for pets are neonics. That I did not know. If you go to chain stores and buy flowers and plants for your gardens, those also have been treated with neonics. So before we get into our interviews, because we've got 3 amazing interviews today, I wanted to do a brief, what exactly is a neonicotinoid? Because we hear about it a lot. Neonics are a systemic pesticide. Did you hear the loon?
Oh, there it is.
That was super early. What systemic means is it's throughout the entire thing, throughout the entire plant. And when originally released, people believed that what you do is you would put this pesticide kind of as a powder on the seed. And then as it was water and growing, the pesticide would go inside the plants. And we kind of thought it only went to the foliage, the leaf of the plants, to protect those leaves from pesticides. But we've since learned it's systemic. It goes everywhere in the plant. It is in the nectar and the pollen and the fruits. It's inside of it. It's part of it. In some ways, that's what makes neonicotinoids so powerful as pesticides, but it's also part of what is causing problems. It's been strongly linked to colony collapse disorder in honeybees, which are canary in the coal mine for a lot of our native bees and other pollinators. If we can easily see the honeybees being affected by them, we know a lot of our native pollinators are being strongly impacted as well. Another thing that makes neonicotinoids so successful and such a large part of our current pesticide management plan in farms is that they're relatively cheap and they're combined with other treatments. So oftentimes, if you're a farmer, you're getting your seeds pre-treated with fungicides and pesticides, and then you don't have to reapply them after planting them. And in some parts of the country, farmers have reported that they maybe don't need a neonicotinoid, but they do need a fungicide. And the only way to get that fungicide is with the neonicotinoid added in as well. That unfortunately makes it being used when it's maybe not needed, and it's used more as a preventative than being used as part of integrated pest management, which we've talked about in the past, which is where you apply a specific tool to a specific job for pests. Now, this does allow us to eat pretty reasonably cheaply. It makes our produce a lot cheaper. It was thought to be safe for vertebrates compared to other pesticides. It's another reason it was really heavily adopted. But as with all things science, new data is discovered, and we have to change our thinking based on data, not how we really want something to be. And so there is growing evidence that it impacts birds directly and indirectly. There's growing evidence that mammals in Minnesota are suffering consequences from being exposed to it. And we know that fruits such as strawberries that are treated with neonics have that pesticide in the fruit, and you can't wash it off because it is inside the fruit. It's part of the fruit. So we're starting to do research to see how is that affecting humans as well. So today, to explore the impact that neonicotinoids are having on Minnesota directly, we're going to be talking to three guests. We're going to talk to Dr. Charlotte Roy from the Minnesota DNR, who has been researching bird and mammal exposure to neonicotinoids in Minnesota. Hardy Kern, Director of Government Relations, Pesticides and Birds campaign at American Bird Conservancy, will talk to us about how the loss of insects from neonics that are not crop pests have impacted bird populations. And finally, neonicotinoids are very water soluble. That's why the sea can take them up. And they've been found in water systems wherever they're used. So to discuss how they're being found in fresh water and how long they lasted in fresh water, we're going to talk to Dr. Bill Arnold from the University of Minnesota. So are you ready, Angela, to explore this?
I'm ready. I'm pretty excited about our first guest because I saw a talk from Charlotte at one of our Wildlife Society summer conferences, and I was just really blown away by the amount of research already showing the effects on wildlife. It was one of those talks you walk away from feeling like, wow, the work we do is really important and bringing that awareness around these. And we have work to do, right? We have some work to do to spread that message and to find better solutions than what we currently are doing. So I'm really excited for Charlotte Roy to join us from the Minnesota Department of Natural Resources. Welcome. Can you introduce yourself to our listeners and a little bit about what you do at the DNR?
Absolutely. And thank you so much for those kind words. Very, very nice. I am a wildlife research scientist with the Minnesota Department of Natural Resources, and I've been doing research for the DNR for 17 years. I started out studying waterfowl, and a little over 10 years ago, I had the opportunity to take another position that focuses on our 4 species of grouse: ruffed grouse, spruce grouse, sharp-tailed grouse, and prairie chickens. So I study a variety of issues that affect these birds and their populations, ranging from habitat loss to management approaches, genetic hybridization, pesticides, and pretty much anything else that could potentially be affecting these birds. So there's a lot of interesting aspects to working with grouse, and I enjoy working in the diversity of habitats used by these 4 species, which include forests, prairies, and pretty much everything in between.
I really wish we had some of those species here in the Twin Cities. We looked for at Crowhassan, but unfortunately it's not big enough. So hopefully people that have explored elsewhere in the state have gone to watch some of these birds. They are just super, super amazing.
I love how you don't realize you're hearing drumming until it's almost over. You're like, oh, there it was. I got chased by a spruce grouse for like a quarter of a mile one time. I was in a whole group of people, but it just zeroed in on me because its chicks were kind of nearby, and it was just pecking at my ankles for as I was running away from it, trying to be like, I'm not hurting your young, leave me alone.
That must have been very terrifying for you.
It was really more funny. I think the terrifying part was I was like 19 and I was kind of embarrassed because everybody made fun of me for being scared of a bird. I'm like, I wasn't scared. I was just trying to keep it from being stressed. So, but yeah, it was fun.
They're definitely cool birds.
I'm curious, why is it important to study if and how wildlife in Minnesota are affected by neonicotinoids, especially things like our grouses in the state?
That's a great question. I'll start at the beginning. Neonicotinoids, or neonics as they're often called, are one of the most widely used classes of pesticides worldwide. They replaced some pretty environmentally persistent pesticides and became widely adopted in the early 1990s. So, as you had mentioned before, they're used as seed treatments, in which they're applied directly to the seeds, in soil and trunk treatments, in foliar sprays, and as well as in other applications and uses. And basically, they're very toxic to invertebrates and at concentrations that are generally considered safe for people and wildlife. However, neonics have received a lot of attention in the media because they're not selectively targeting invertebrate pests and can harm other invertebrates such as pollinators, which are important for food production, and aquatic invertebrates, which are important food sources for wildlife. So, a lot of the attention has focused on bees and other pollinators, which have been experiencing declines. However, at high concentrations, neonics can be harmful to birds and mammals too. Some of the ways in which birds and mammals might be exposed to high doses of neonics include eating those treated seeds or eating recently sprayed foliage, for example. So, one of our studies quantified how commonly those treated seeds are available on the soil surface during spring planting. And we found that treated seeds such as corn, soybean, and wheat, and various other crop types can be available to wildlife as a result of spills during seed transport to fields or when refilling equipment during planting. Sometimes seeds are incompletely drilled into the soil during planting, or they can be exposed by digging. So these seeds on the soil surface can be eaten by wildlife, and the concentrations of neonics on these treated seeds are quite high and often high enough to be causing sublethal effects.
And you actually did some studies in Minnesota to see are animals actually eating these spilled seeds? How did you figure out if they were, if they weren't?
Yeah, so we placed trail cameras at simulated seed spills, and we basically look to see if wildlife are eating the seeds. First, were they finding them? Were they eating them? And or were they deterred by the weird colors? Because these treated seeds often are brightly colored by law so that people don't accidentally feed them to livestock. So they look weird. They're, pink and purple and green and all different colors. And so some folks thought maybe that would deter wildlife from eating them, or perhaps the taste or another factor. But we actually found that at these spills where we placed trail cameras, lots and lots of wildlife were consuming the seeds, including various blackbirds, crows, sparrows, pheasants, turkeys, mice, voles, deer, squirrels, rabbits, raccoons, red foxes, and more. So quite a lot. Not just one or two species, but quite a lot. Sometimes the animals would discover the spill and not consume right away. Other times they consumed just a little bit, but maybe not enough to cause a problem. But there were many instances in which we observed wildlife eating seeds that we would expect to produce adverse effects on survival or reproduction based on studies that have been done in lab settings. So this indicates that there's potential there for these seeds to be harmful to wildlife and their populations.
I'm curious if you know, are they actually more attracted to the bright colors than a standard seed, or has research not been done on that yet?
Or maybe easier to find.
We have not done that research ourselves. I know there's been a few studies where they've put treated seeds nearer to cover than untreated seeds, and the birds will select the seeds that are closer to cover where they can escape from predators. I believe those studies were done in Europe. So I think when you do a side by side test, like in the lab, the birds are going to preferentially take the seeds that are not treated. They are thought to cause a little bit of, I guess, for lack of a better word, maybe indigestion or some kind of upset, stomach upset after being consumed. We did find them eating seeds, the wildlife eating seeds, and some wildlife are eating quite a lot of seeds.
And do they have to eat a lot of seeds for the health effects? Are we talking like they eat one or two seeds and that's it, or do they have to eat dozens and dozens of seeds?
That is a very good question. And the answer is it depends. It depends by species. It depends based on whether they're, eating other things before they eat the neonics. Is it the only thing they ate that day, or is it, have they eaten something else that's being mixed in there? It depends on how the bird or mammal metabolizes the neonics and excretes it. So the body basically takes these chemicals and tries to get rid of them. There's a lot of different things that influence how much it takes. Generally, the bigger the animal, the more it can tolerate in terms of absolute concentrations. but bigger animals also eat more. So you would expect there to be some relationship there.
I wanted to add in because this was something I remember from your talk at the Wildlife Society was about in the lab studies were actually hard to do because of the lack of non-treated seeds to use in a control. And I'm wondering
Yeah.
if you can answer more of like, these are purposely simulated spills, but how likely is it for wildlife to come across these spills or neonic seeds?
Yeah, we actually looked at that in one of our studies, and we quantified that on the landscape. So we essentially drove roads in the spring, and we looked for seed spills, which are quite visible from a distance because they're brightly colored. They pretty much are beacons when you're looking. If there's a large spill, you can see it because it's, kind of brightly colored. And we found a really large number of spills in Minnesota during the spring planting season. And we also walked fields in the spring after planting to see if there were seeds that were not completely drilled below the soil surface. And we did find that there were quite a few seeds available on the soil surface, and it did vary by crop type. So corn is drilled deeper than like soybeans and wheat. So we found more treated soybean and wheat seeds on the soil surface than corn, which is drilled deeper to promote the proper development of the plant. So it did vary by crop type. But yeah, we were rather shocked at how frequently these spills are available to wildlife.
Did you did your studies provide any insights on how to prevent or minimize exposure of these seeds for wildlife?
Yeah, yeah. As a result of our study, we actually had numerous conversations with the seed companies and with farmers to try to understand why these seed spills were occurring and maybe try to identify some solutions. And in our conversations, the seed companies expressed that they didn't realize these spills were occurring and certainly didn't factor that into their recommended use. When we talk to farmers, they express that, they really enjoy viewing wildlife. They don't want to harm wildlife, and they didn't realize that the seeds were actually harmful to wildlife. So they hadn't really been cleaning them up or covering them or anything like that. And moreover, they hadn't really noticed the safety warnings on the back of the seed bags that indicate these are harmful to wildlife, but also indicate you should wear personal protective equipment when handling these seeds. So if they weren't protecting themselves, then it's very, it's not a leap of faith to believe that they also didn't realize that it was a problem for wildlife. So we talked about that a little bit and possible solutions. And one solution, of course, is to cover up the spills with soil. It's not really easy to pick them up, and you can't really put them into the equipment with all that soil and stuff like that. But you could cover them up, and that would make them, less available to wildlife to eat. And we essentially tried to promote a campaign to spread the word about covering up seed spills. And so that was largely led by quite a few of the seed companies because they were, very bothered that this was occurring. They didn't factor that into any kind of safety considerations. And so they made an effort to get the word out. And then I went and I spoke to seed applicators throughout Minnesota to try to also get the word out that, these seeds are harmful to wildlife, and we need to be responsible stewards of seeds so that way they don't harm wildlife. And I hope it made a difference. But other possible ways that exposure could be reduced is if people only use them when they actually have a problem. So if you don't have a pest issue, then maybe you don't need to be using neonics every year. Canada is taking this approach now, and it's really reduced the amount of neonics that are being used on the landscape. And because neonics are water soluble and can be transported far away from the site of application through water, they are able to move long distances. And so one study had indicated that only 2 to 20% of the seed treatment is actually taken up by the plant as it's growing. So that means that 80 to 98% of the treatment not taken up by the plant. So that's a pretty shocking amount of chemical that is
Where's it
not
going?
going up into the plant. Exactly, exactly. But at a more personal level, you can purchase food, flowers, and greenhouse plants that have not been grown with neonics. You can purchase organic food. You can appeal to your legislators for the changes that you want to see. Citizens are allowed to comment on registration of new and existing products by the Environmental Protection Agency as well.
When I was doing a lot of reading to get ready for this, it seemed like a lot of research is showing that neonics are thought to save money because of the pesticide application issues for farmers, but it's actually costing them money because like 80-90% of the time it's treating a problem that doesn't yet exist. And so there was a lot of research pushing for don't apply it every year, only apply it when it's maybe needed. Maybe even applying it later in the year might be better because starting right out as a seed, it's most active when some of the pests they're trying to get rid of aren't even active yet. You also, as part of your studies, studied how neonics are processed in the body with captive chickens. How did you do that, and why did you do it, and what did you find?
Yeah, as part of an earlier study funded by the Environment and Natural Resources Trust Fund and the Minnesota Department of Natural Resources, we did an experiment at the University of Minnesota in which we dosed captive domestic chickens with imidacloprid, which is one of the more toxic of the neonics to birds in particular. It's commonly used as a seed treatment on things like soybean and wheat. So we used that one to study the effects on birds and how the chemicals handled by the body. Grouse don't tolerate captivity very well, so domestic chickens were a very good surrogate. They're closely related taxonomically, and so we expected their physiology to be very similar in the way that their bodies would process the chemical. And so we started with 3 dosing levels, 1, 5, and 20% of the median lethal dose, which is the dose that's expected to kill about 50% of your test subjects. So we took 20% of that value as our high dose. But unfortunately, we had to drop that group because after the first treatment, we had birds that were twitching or unresponsive, and so we needed to euthanize those birds, and so we lowered our dose group to 10%, and basically we exposed them once a day for 7 days, and then we were collecting blood and feces to see if we could detect the chemical in the blood or in the feces. And then after a couple weeks we actually euthanized the birds so that we could evaluate how much chemical was in their different organs. Now, it's important to point out that neonics do not bioaccumulate like a lot of other pesticides. So they aren't stored by the body for a long time like a lot of other pesticides that you hear about, but rather they are excreted by the body. So the body finds a way to get rid of them pretty quickly. Essentially, what we learned is that we can detect the chemicals in the feces for about 15 days, or at least 15 days after exposure. We could find it in the tissues at some doses, but not at others after 15 days. And in the blood, it disappears really quickly, like it was not detectable in any of our birds within a couple of days. So this gives us some insights into if we're going to try to detect exposure in the wild, what kind of time frame we need to be working in to be able to time that sampling with periods that we expect exposure to be occurring. Because if you wait too long after exposure and there aren't repeated exposures or continual exposures, it's going to look like that bird wasn't exposed when maybe it was. And so by doing this work, we learned that for that we can collect samples without killing birds by using feces, and that we need to sample within a few weeks of when we expect the birds to be exposed to the chemicals, or at least if we sample after that time frame, we might not detect an exposure that occurred. And we also learned that, by lethally sampling within that a short time frame after exposure, we could detect the neonics in various tissues. So, as you go forward and try to apply these types of concepts to wild populations, you just need to be cognizant that what you're detecting isn't necessarily going to tell you how much that bird was actually exposed to, because it's kind of a moving target, right? It's continually changing as the body is excreting it. And just because you don't detect it doesn't mean that bird or critter wasn't exposed at a previous time point and has now eliminated all of the chemical.
When I read that, I would have thought it not bioaccumulating would have meant that, oh, the body gets rid of it, it's maybe less toxic. But it sounds like if you had to drop from a 20% down to a 10% dose, it can still be pretty toxic. Is that correct?
Yeah, at high concentrations, sublethal effects have been reported in a variety of species. And in our trail camera study, there were a number of different birds and mammals that were eating enough seeds that you would expect them to be experiencing some sublethal effects. So some of those sublethal effects include things like adverse effects on reproduction. So for birds, that would include later clutch initiation, lower hatching success. Eggshell thinning has been reported. You brought up Rachel Carson earlier, and when I hear eggshell thinning, it harkens back to the days in the 70s when they were discovering some of these really terrible effects of different pesticides at the time. But these are things that are documented in birds. Then in mammals, there's also a slew of adverse effects that occur, things like low birth weight in young, or basically stillbirths and things like that. So it's a pretty wide set of sublethal effects that can be experienced.
That's really interesting. Maybe I'm misinterpreting, so correct me if I am, but it sounds like maybe some of our initial thoughts that, well, this really isn't that toxic for mammals or for birds or for vertebrates is correct. It sounds like maybe it's more toxic than we initially knew.
Oh, well, it's nuanced, right? So at low doses, it's considered safe for birds and mammals. And that's why it's used on food that humans eat, right? Because it gets taken up by those plants and we're exposed to very low levels. And so, the regulatory agencies have deemed that those exposure levels are safe for humans and for use in these food products. But when you get exposed to the high concentrations that are on a seed, that's a very different level. So that is where we start seeing those adverse effects, is when we're encountering those really high unintended concentrations. Nobody planned for birds and mammals to eat these seeds when they produced them. They were supposed to be, the chemicals were supposed to be taken up by the plants, and then the plant was supposed to be protected from invertebrates. And invertebrates have very, very low thresholds where you start seeing, adverse effects in invertebrates, but vertebrates are many, many times higher than that. And so what we get in the plant that's grown is not expected to even be close to this threshold.
That makes sense. And that's what we were judging on. But all technology has unintended consequences, and pesticides I would consider technology. So interesting.
Yeah,
Um,
yeah.
is there anything else that you want us to know about your research or your studies on neonicotinoids?
I think that we need to be mindful that as we're, when we initially adopt products, we try to do a little bit of research. Often that research is done by the person proposing the product or the company proposing a product. And so it's not necessarily done by people that are not vested in the outcome. And so I think it's healthy for us to have, a lot of research on the front end, but at the very least, we need to be mindful that we can't think about every possible um, outcome at the onset. And I think it's important for us to revisit things along the way. And I know the EPA has been revisiting their registration and doing a registration review for these chemicals for a number of years now. And so I think they're intending to make some changes, but they certainly, at least from what I could gather from their draft document, it doesn't look like they're going to be banning these seeds or anything to that effect. So I think we just need to think about things a little bit more carefully and continue to revisit them to make sure that we really understand the full impact.
That makes sense. As we realize consequences we're going to test for, keep our minds open, use the data to make decisions, and be willing to adjust. Do you have any advice for listeners who want to enter a scientific career?
Oh gosh, that's a good question. I would say, pursue what interests you, but have realistic expectations about what a career in a particular field actually means. So, loving the outdoors, for example, doesn't mean that you get to spend all day outdoors every day in a natural resources career. So talk to other people in your chosen field about what their day to day is really like, and set realistic expectations of what it means to have a career in that field. And maybe you can do what you love by pursuing a career that gives you more free time to do it. But if you can match what you love and your career, you never work a day in your life.
I like that. That's awesome. Thank you. Thanks so much for talking to us about your research on how vertebrates in Minnesota are being exposed to neonicotinoids. And it was just really, really fascinating. I really appreciate it.
Thank you for having me. I enjoyed being here.
Next time, we will talk to Hardy Kern about the loss of non-target insects from neonicotinoids and its impact on birds.